Hello, my name is Adrian Santiago, and today I will be talking about the project that I have been working on, which is the potential link between cell swelling and cytoskeletal rearrangement. Astrocytes are specialized glial cells that are seen throughout the central nervous system and are very essential to the maintenance of neurons. Neurons are information messengers. They use electrical impulses and chemical signals to transmit information between different areas of the brain and the rest of the nervous system. Astrocytes support neurons by releasing an antioxidant known as glutathione. Glutamate, a glutathione precursor, can also cause fatal damages to neurons when released at high levels during a stroke. During a stroke, cells swell, causing activation of the volume-regulated anion channels, VRAC, which leads to the release of glutamate, contributing to neuronal death. The exact mechanism of VRAC activation is unknown. In this experiment, we hypothesized that when a cell swells, this may cause cytoskeletal rearrangement, which can be a leading factor in the increase of VRAC activation. To test this hypothesis, we used a chick embryo as a model to study the astrocytes and their cytoskeleton. We dissected the optic tectum of the chick embryo from their embryonic day 8. We then cultured them for long periods of time and were then treated with the hypoosmotic treatment, which induced cell swelling. We then used phalloidin staining to visualize the actin fibers of the cell. So as you can see in figure 1, it shows the astrocytes at both a low and high magnification in a normal environment with no treatment but media. We were able to see the organized patterns of the straight parallel actin fibers throughout the entire cell. However, on figure 2, it also shows the astrocytes at both a low and high magnification. However, in this figure, the astrocytes are shown in a hypoosmotic environment. As you can clearly see in image G, it shows a more scattered organizations of the actin fibers. Based on these results, we have concluded that the induced cell swelling did affect the rearrangements of the cytoskeleton in the astrocyte. Further experiments would have to be done to completely understand the mechanisms of how VRAC is activated. This project has shown that there are changes in the cytoskeleton when the cell goes through a hypoosmotic environment, which depicts cell swelling during a stroke. Although previous groups have explored the relationship between actin reorganization and cell swelling, we aim to determine if this actin rearrangement influences cell signaling pathways known to regulate VRAC activation. Thanks for listening.